Hello, my name is Nicolás Morato. I'm a Ph.D. candidate at Aston Labs at Purdue University under the direction of Professor Graham Cooks. And today I want to talk to you a little bit about this novel high-throughput technology that we've been developing for the past 6 or 7 years, which is based on Desorption Electrospray Ionization mass spectrometry, or DESI-MS, which we think has a lot of applications, particularly for reaction screening, nanoscale synthesis, and bioanalysis. So to start really talking about what this platform can do and how it works, I think first we need to set some concepts straight. And the first one is, okay, we're saying that this platform is based on DESI, Desorption Electrospray Ionization. So what is DESI? Well, DESI is an ambient ionization method, and I will get what that means in a minute. But it basically works by using a charged solvent to spray that impacts on a surface, and then your sample is deposited on this surface, or your surface is your sample. Then there's a microextraction event occurring, and splash microdroplets, as you see in the GIF on the bottom left of the video, are splashed, and those are the microdroplets that carry the desorbed analytes to the mass spectrometer. So that involves this microextraction event and involves these secondary microdroplets, and those are two quite important points for later in this talk. So I mentioned that DESI was an ambient ionization method. What the hell does this mean? Well, this means that you can perform analysis of samples directly in the open air without or with very minimal sample preparation. You don't have to do ionization in vacuum, and you retain, of course, all the capabilities of MS without having to have your ionization happening through, for example, lines of solvent going around or happening inside a vacuum chamber using a laser, for example. And DESI was the first of ionization methods, and currently you can find many of them, including vapor spray or DART, which was created about the same time as DESI. And of course, many more are to come. So if you have heard anything about DESI, and if you have not, that's okay. But since its inception, and if you've heard about it, you've probably known DESI because of tissue imaging or imaging, chemical imaging in general. So since DESI started, here you see some reference 2008 or something. DESI has been used to image samples, in particular tissue sections, but also others like fingerprints, like you here see in this example, where you don't only keep the distribution, the profile of the fingerprint, but you can also get chemical composition. For example, here I think it was a person that touched cocaine, and you can see cocaine from the fingerprint. But the question now is, how do you take this imaging capabilities, which is what DESI is mostly known for, and translate it to high throughput kind of workflow? And the answer is, in fact, simple. If you think about tissue section or a fingerprint in its most abstract way, you will think about just chemicals, different mixtures of chemicals with different compositions that then you're having certain special distribution across the tissue, across the fingerprint. So mixtures of compounds with certain special distribution. You can make that too in the lab. If you make your own array of mixture of compounds with a special distribution that you define, you can apply the same principles of doing imaging to perform high throughput analysis. And that's how you obtain some of these figures here on the bottom right of the corner, where you're imaging a generated array of reaction mixtures, and you're getting, for example, where you generated your products of these reactions. And you can apply this not only to reaction mixtures, but you can create an array with biofluids, you can create an array with biological assay samples, and so on and so forth. But to actually convert the imaging idea to the high throughput experiment, you have to take into account some other couple of factors. The first one is that the imaging experiment usually involves rastering, because if you want to acquire a high resolution image of sample, say a tissue section, you're going to have to raster across the tissue section, making lines, and then making those lines close to each other, because your resolution will depend on how fast you're going across each line and how close you can put those two lines together. The closer you can put them, the slower you go, higher resolution. They're all great. But for the high-throughput experiment, you actually don't care about the resolution. You care about going fast. Usually, those two go in opposite directions. If you look at the picture shown on the screen right now, you will see that if you do the rastering experiment on an array of samples, you're being very inefficient. You're spending a lot of time over white space, basically over space that has no samples, and you're rastering the same sample several times. So you're getting redundant information. Besides, if now you imagine that you don't put all these samples, but you just put half of them, well, you're going to take the exact same amount of time, which is also not very efficient, not very logical. So this is not a good way of doing it, or it's not the best way of doing it. So what do we do? Well, we can take now the positions of this array that we created, because we created, so we know that the positions are fixed within the array. We just need to calibrate the corners of the array. With that, you can create a virtual grid, and you can just move through any of the positions on the grid. You don't have to go to all of them. You can select which ones to go. And by calibrating these positions and selecting to which of the spots you want to jump, you can do something like the GIF it's showing now on the slide, and you can jump from sample to sample, spending about half of a second per sample. That is the throughput that we work currently, but half of a second per sample before jumping to the next one. And this gives you the flexibility of analyzing whatever number of samples you want, just a subsection of all the ones that you put in, or all of them, and you waste very minimal space in jumping compared to rastering, and you're actually investing most of your time trying to do the analysis. And this allows you to achieve throughputs better than one sample per second, which is the rate at which we currently operate. There's another important factor, and this is mostly applicable to reaction screening, and is that if you are screening organic reactions, you're typically going to have to mix your reagents and then incubate them or heat them or whatever it is, but you have to waste some time before you actually read the products of these reactions. If you think about a round bottom flask reaction, you're going to have to put it on a heating block for hours overnight, whatever it is, to then see the reaction the next day to find the products. But that limits a little bit of your throughput, right? Despite you can do this in batch and so on, it limits a little bit of your throughput. Here we have another factor that is quite important, and is that we can do the screening of accelerated reactions. And this has to be to a phenomenon that occurs in those microdroplets that I referred to at the beginning that generate from the DESI event. And in those microdroplets, because of the high surface to volume ratio, that surface has very particular properties. So if you put, for example, a reagent in the DESI spray and another reagent in the surface, or you put both on the surface, it doesn't matter, they will get mixed in these droplets. And when you see at the end, you will see the product of those reactions, either if they just deposit here on the surface or if these droplets are sampled from the mass spectrometer. And this occurs in milliseconds. These droplets have millisecond samplings. So you're seeing the product of reactions very, very, very fast. And this is due to this particular surface of the droplets in which you have an interface between the gas phase and the solution phase. And one of the main hypotheses of why this acceleration happens is basically because of the particularity of this interface. So you're kind of on an in-between state between a solution phase reaction, where you have to overcome this huge energy barrier, as you see in the green line, and the gas phase reaction, which you basically go downhill, and then upon every effective collision, you get a reaction. So you're somewhere in between where your reactions can get accelerated by several orders of magnitude, up to probably 1 million. That is the distance between the fastest solution phase and the gas phase reactions. And of course, there are other particular phenomena at interfaces that become quite important. For example, you can have high electric fields at the interfaces and generate reactive species, radicals, but also you have extreme pHs, which can protonate things that, for example, cannot be protonated or wouldn't be protonated, generally speaking. For example, CO2 that can get protonated and then be abstracted to, for example, capture CO2 as carbonic acid using just droplets of containing amines. And if you put all these concepts together, the idea of DESI translating the imaging experiment to the high-throughput experiment, the idea of going spot to spot, the idea of having accelerated reactions, then you can consolidate the platform that we have and use today. So on the slide right now, you see the processes and the instrumentation involved on the high-throughput system in its current version. So first, we have a fluid handling robot in which we can use to automatically prepare the samples. Let's say that it's organic reactions or that it's biological assays, whatever it is. And we can prepare them in any format if they need to be, for example, incubated, enzymatic reactions, or whatever it is. But at the end, we want to end up with a 384 well plate. And in this 384 well plate, we use it because we have a 384 pin tool. This pin tool has a 50 nanoliter, it's calibrated for 50 nanoliter transfers. So we are using just very, very small nanoliter aliquots to generate high density arrays from the 384 well plates to what we call a DESI plate. This DESI plate is just a glass slide coated with a PTFE membrane, which is what you see, that white rectangle with some dots in the middle of the screen. And in this DESI plate, we can spot, using the pin tool, up to 16 different 384 well plates or replicates of the same one, making high density arrays with 6,144 samples per plate, or any multiples of 384 that you want. You don't have to put 6,000 if you don't want to. So these arrays with nanoliter samples are then transferred automatically from the robot to the DESI stage of a mass spectrometer, and we've coupled two different systems to the overall high throughput platform. And in one of them, you see a black box with some bottles around that is a flow switch matrix, which acts as a solvent delivery system. So you can change your DESI solvent in between plates, or you can create gradients across plates, and so on and so forth. Then once you get to the DESI stage, basically the analysis occurs automatically. You move across, after calibrating the positions of the spots, you just move across, jumping sample to sample, spending less than half of a second on each sample before jumping to the next one, with overall throughputs better than 1 sample per second. And in those secondary droplets of the DESI, you can have these accelerated organic reactions, as is denoted in the orange rectangle. Eventually, once you finish your plate, you have a huge amount of spectral data that you have to process, and when the user starts this whole workflow from the fluid handling workstation, it also inputs a plate layout, which basically contains, okay, what did you put in, what are you looking for, which M over Z values are you interested on each one of the wells, on each one of the samples. So using that user input and the raw mass spectral data, you process everything together and generate a peak list, which associate the information that the user requires for each one of the samples. And with that, you can generate interactive heat maps or any other sort of output. This is more tailored to whatever you want to do. For example, if you're doing quantitation, or if you're doing some enzyme kinetics, we have automated code to do all this from the mass spectral data. Now from this, let me show you how it works in real life with this. Now let me show you a video of how it happens. So here first we're seeing the deck of the fluid handling workstation. We're taking the pin tool dips on a well plate, and then it transferred to the DESI plate, which is that white thing, is a glass slide coated with a PTFE membrane. Then you take the DESI plate that is just supported on a 3D printed holder, but it's easier for the robot to grab. You will put it in a shuttle, and then it will send it to the back of the robot, where you will take the slide again, and then put it on the DESI stage for the analysis to start. Now we're seeing the screen of the computer that is connected to the mass spectrometer. So first step is going to just be calibrating the positions of all these samples, and this can be done automatically with image recognition software, but here it's done manually. We go through the corners, we know all the positions of our array, then we start. As you see now, the stage is moving underneath the spray. Some spots have some color, and you can see them moving. Then here you see live mass spectral data acquisition, all the spectra that are coming from the different samples. We're spending more or less half of a second in each spot before jumping to the next one, so the overall throughput is better than 1 sample per second. And of course, it's dependent on the density of the spots you have. The more spots you have, the less distance you have to jump, so you can go a little faster overall. But we're spending about half a second in each spot, requiring 3-4 mass spectral scans. Then here you see on the right that you generate this interactive heat map, basically in real time with the data acquisition system. If you click any of these particular spots, you will see the mass spectra, the corresponding mass spectra, and then you can filter by intensities, by the type of reaction you had, and so on, like we're doing here right now. We filter by reagent. So these are the key concepts. This is how the platform works. Now, what have we done with this platform? What are the applications? Well, what we think are the main pillars of applications of the system is basically the three key steps of a drug discovery workflow. You want to screen reactions to generate molecules. In here, let's call them candidates. Generate these molecules, and then you want to be able to synthesize these molecules in amounts enough that you can test them. And you can assess their bioactivities, and then whichever compounds you find which stronger bioactivity, then you just basically generate analogs, iterating back over this cycle. So we think this platform can consolidate this whole drug discovery workflow in a single system. Let me try to show you how. So we focus on the first step, which is screening. Now let's see what we've done. Well, we've screened several organic reaction types. Here you see two examples of results obtained for the Suzuki cross-coupling. There are many, many, many more that we've explored, and it just shows you the diversity of chemistry that you can study. We can study, for example, the reactions mechanisms itself, see what is the influence of the stoichiometry, the orders of addition, the different ligands, solvents, catalysts, and so on. We can optimize a particular transformation to find the optimal synthetic conditions, or we can just assess in general reactivity trends. And because I told you about this microdroplet accelerated phenomenon, one of the main questions that we get is, okay, is this comparable? So what you get with DESI, that you say that you can go really fast, going less than 1 second per sample, and having these reactions in droplets and so on, does this really compare with what you obtain with a normal screening? And the answer is yes. So if you compare the DESI results with LC-MS results, you will see that the trends are in general pretty much comparable, but what you get with DESI is that you save orders of magnitude in terms of time and solvent consumption, as you will see in the bottom of the screen. Of course, these heat maps are on the same intensity scale. You see that the DESI intensity is a little lower, but that is known. DESI gives you a little lower ion intensity than LC-MS, but the trends are pretty much identical. And another question will be, okay, it does work, but do your reactions in droplets into this super fast platform really translate to scale-up conditions? For example, flow synthesis. The answer is yes, too. So we, for example, explore the synthesis of Lomustine, and in particular the nitrization step that you see in the reaction scheme at the top of the slide. We can explore different solvents, different nitrization agents, and so on and so forth. And the optimal conditions found using the DESI screening were translated, amongst others, to compare to a flow synthesis setup, and surprise, the conditions that were found optimal with the DESI-MS system were also found optimal for Lomustine synthesis in the flow setup, providing milligrams per hour Lomustine with high yields. Now, we've recently also tried to explore other types of reactions. Instead of taking simple building blocks, try to derivatize already active and complex molecules, because this takes you closer to the kind of drug discovery workflow in which you find active molecules and you generate analogs and you have to test them again. So in this case, we've demonstrated with, for example, Ezetimibe, which is a medication used to treat high cholesterol. And we've tried to derivatize the phenolic group of Ezetimibe using several reactions, including alkylation or, for example, this azo-like chemistry. So that is the reaction that you're seeing on the bottom of the screen. And as you see, we found this reaction being very dependent on the solvent composition, the stoichiometry of the reaction, which are the numbers you see below the heat map, and the azo compound that you're using for the conjugation, as you also can see from the heat map. On the right, you see the full mass spectrum of the reaction, in which you see that the main peaks are still the non-functionalized Ezetimibe, but you can identify the sodium adduct of the azo-like conjugated molecule that you can also characterize through tandem mass spectrometry. And that is another of the great capabilities of the platform, that if, for example, you have something that you don't know what it is, or you want to confirm the structure, you can just go back after the whole screening, go back to that same spot, and then acquire MS/MS data with that same little amount of sample that you put in. And I think I forgot to mention, but the pin tool that we use is calibrated for 50 nanoliters. So we're using 50 nanoliter samples to perform the analysis that typically translate to low nanograms down to picograms or even lower of sample. with very, very good results because it allows you to both do full MS and do MS. And then you can also, instead of doing a single transformation, you can explore all different reactions that you want. So this is in a single experiment. I think we explore reactivity trends across 22 or so reactions. On the left, you see some example mass spectra of this reaction. So if you see the bottom, you will see mostly what is left is the base that we use for the reaction. Besides of that is all products, so the reaction seems complete. And on the top, you will see that you still have a lot of reactant left over, that's cyclohexylamine, and you have two peaks. One corresponds to the product, 235, and a 233 that we don't know what it is. So how can we figure it out? Well, as I told you, we can do MS to characterize these unexpected products. And this is an example of the same reaction, just I think different reagents. The final product expected is 221, but you see another unexpected peak at 219. So what is this? Well, if you think about the mechanism of reaction, it's easy to know. It's just imine, the non-reduced intermediate towards the amine. But if you don't know, you can take both of them, the 221 and the 219, and characterize them so that in the top on B and D, you see both the MS of the imine and the amine respectively, and you see that they have very similar fragmentation patterns, basically shifted by 2 units, and they correspond to normal radical losses induced by the nitro aromatic group. Basically, you lose radical N2O, radical NO followed by CO. But then you generate these radical species that only in the amine have also the loss of molecular hydrogen from each one of these fragments, from the radical cations generated. So that's how you can distinguish them both, and you can take any of these ions and do MS3, for example, and continue your further characterization if you didn't know what these species were. You can take this to extreme. Now, what if you want to acquire spectral libraries? Because that's very useful if you want to do metabolomics or you want to do untargeted identification of compounds and so on. But you need these libraries to be able to compare a spectrum. And usually those are tedious to make because it will take forever. But what we did is take 20 and something thousand compound library, put them on the plates, and then screen them, acquire MS spectra from all of them very efficiently. We're spending just about 5 seconds on each sample. And in this way, you obtain high quality spectra, as you see on the bottom right of the screen. And you obtain high density information from all these molecules, which is consolidated in the histogram, and the 2D MS plot, which is this blue square that has on the axis the product M over C values and the precursor M over C values, in which you can start identifying trends where diagonal lines correspond to common neutral losses, the fragment that is lost without charge after fragmentation of an ion, and then common product ions, which are the horizontal lines across. And so with all this information, of course, you could fit machine learning algorithms for structural elucidation. Now, the second step on this cycle is the synthesis. And I'll just show you one slide about the synthesis, which is kind of the concept behind it. We've talked about screening organic reactions. We've talked about having these accelerated reactions in the droplets. But then, how we're going to do synthesis with the whole set? Well, what if instead of sampling those droplets with the mass spectrometer, that is what we've shown in the past couple of slides, what if you put something in between and collect the droplets? If these droplets have the products of accelerated reactions, then at the end of the day, that's what you want. So you can collect those products and then use that collected material directly to perform a bioassay, because you're collecting nanogram amounts of material. But that is enough to test biological activity in many or most of the cases. So what we've used currently is just paper. Put paper in between, collect the products, deserve the paper, and test it. And so this step links the reaction screening. You're going to collect the products of successful reactions that you deem successful by the MS profile, that you characterize with the online MS, etc., etc. And then you take those materials, collect them, and put them on the bioassay. And the question is how we're going to do the bioassay. Well, we're going to do it exactly with the same platform, exactly with the same workflow. But the question is, okay, why don't we use conventional kind of bioassay strategies, right? everyone uses plate readers. Why don't we just take this and throw it on a plate reader, which is already done, and there are commercial kits and so on. Well, the thing about plate readers and most commonly done high-throughput bioassays is that they involve this optical or radiometric readout. But most biological systems involve no color, fluorescence, or naturally radiolabeled species. That means that you're going to have to change your systems. You're going to have to modify your substrate. You're going to have to label them. You're going to have to couple secondary reactions that give you detectable outputs. And this limits the versatility of these assays. You're going to have to start from scratch every time you develop a new one, every time you change the enzyme, every time you change the substrate. And also introduce more work, more expenses, even safety concerns to this assay. And furthermore, it introduces interferences. So what do we want to do? Well, why don't we use a label-free, inherently label-free method as mass spectrometry, and we take the advantages of DESI, which now it's allowing us to sample really, really fast. And because of this microextraction process that I mentioned at the beginning, it has very high tolerance to complex mixtures, including buffers, salts, and detergents, and so on, which is what you will typically put in a bioassay and what will be typically detrimental to a normal kind of ESI mass spectrometry analysis. So how do the bioassays work? Basically in the exact same way, the exact same workflow. With the bioassays, incubate them, coincide, transfer 50 nanoliters with the pin tool, generate high-density arrays with up to 6,144 samples per plate, perform a spectral analysis, and do automated data processing. There is absolutely no sample work and no purification, no separation involved anywhere. And of course, it has advantages of being completely label-free. You can use the native systems of a study as they are. You don't have to change anything of the actual reaction you want to study. Being very fast in terms of the screening, super simple. You can use the same workflow to any system you want to study. That also increases the versatility. And what we really like to emphasize is that it gives you excellent quantitative performance, which is something that people don't usually think about ambient ionization techniques, but we obtained relative standard deviations typically below 10% in most of the determinations we do. And because we have such a high density of spots available, we can do multiple replicates of a sample to actually get an average value if that's needed. So how does this typically look? Well, let me show you here an example of acetylcholinesterase, an important enzyme for the nervous system. If you put different fractions of acetylcholine, different time points of the acetylcholinesterase reaction, you will see that at the beginning everything is acetylcholine, and as you move down in time, going down in this line down, you will see that it turns from 146, that is acetylcholine, to 104, which is choline. And with this information, you can just plot the ratio of these ions over time, and then monitor the experimental progression of the reaction, which you can then calibrate, and estimate initial reaction velocities, do Michaelis-Menten analysis, find all the kinetic parameters that you want. In this Michaelis-Menten plot, behind there are many reactions progress curves with many replicates each. So we have more than 2,000 samples on this plot, but it just takes about 30 minutes to do it with a high-throughput system. And the parameters that you get agree pretty well with what you find reported on the literature. Then you can also screen inhibitors, for example, and we develop a workflow to screen up to 384 inhibitors in just 15 minutes. Of course, you can screen way more, just take a little bit more time. You can take any of these active inhibitors and then characterize it further, for example, through those response curves, as you see on the right, to estimate IC50 curves, to estimate IC50 values, or through Dixon plots to estimate inhibition modes and inhibition constants, and so on. Of course, these Dixon plots involve that you change both the substrate and inhibitor concentration over time. They require a lot of samples. This particular plot has more than 4,000 samples behind, which is something not as scalable to do if you don't have a high-throughput technology. And lastly, what we also demonstrated with the system is that you can do even reactivation. So this enzyme gets inhibited by chemical warfare agents, and using chemical warfare agent simulants, you can simply inhibit the reaction and then reactivate it using commercial oxygens. And this is something that traditional methods cannot do, because the traditional methods based on using thiocholine instead of acetylcholine, and thiocholine is quite susceptible to be broken apart by the oxygen itself. So your readout comes from, who knows, the reaction that is the enzyme that is being reactivated, or the oxygen that is breaking your substrate. Whereas we, because we're using the native substrate, we have absolutely no breakup of acetylcholine, and we can monitor these reactions for hours at multiple different concentrations of oxygens, which limits quite a bit the normal typical assays. I think I run out of time, so I'll just let these slides on the PDF, but this is the latest case that we've studied is on this reference here on the bottom of the corner, and it's related to prostate cancer-related enzyme. So cholesterol sulfate accumulates in prostate cancer, and that's something that was found with DESI-MS. And then this implies that not only you accumulate the metabolite, but also it has been found that you accumulate and overexpress the enzyme that produces it. And if you knock down this enzyme, you can induce death of the cancer cells. But there was no really a way to study this enzyme. So some collaborators developed a fluorescent method with some limitations, including needed an initial incubation, being limited by the purity of the reactant, including having a lot of competitive interferences between the coupled enzyme and the actual enzyme you want to study. But we developed a completely label-free method using the high-throughput DESI-MS technology that gives you comparable results to those of the fluorescent method, as you see here, especially in terms of affinities, finding that this enzyme has way higher affinity for pregnenolone than DHA than cholesterol, contrary to what it was found before. And we've been able to identify that inhibitors that were thought to be good inhibitors with this coupled fluorescent enzyme assay are actually not very good inhibitors for the enzyme that you want, but are good inhibitors for the other one. And so now using this method, we're trying to find inhibitors for the actual enzyme that interests us regarding the prostate cancer treatment. And with that, I'd just like to finish reminding you of the three pillars of the DESI-MS system, which involve high-throughput screening of organic reactions, nanoscale synthesis collecting disruptors that carry the products of accelerated reactions, and then testing them online with high-throughput label-free biological analysis that we've demonstrated in several biological systems, including enzymes, but also receptors, which I didn't show in these slides. And thank you so much for listening to this presentation. I would just like to thank all the people that funded us, our research in the lab, and also personally me, like Isman, and the Division of Analytical Chemistry of the ACS, and all of our collaborators and the people that have made this possible throughout the years, including my lab members. Thank you so much.